I am Dr. K.AMUDHA from Biochemistry department. In today's topic, we can see what is good laboratory practice. That is GLP. Good laboratory practice is an FDA regulation, and it is a formal regulation that was created by the FDA, that is United States Food and Drug Administration in 1978. When it comes for the definition of GLP embodies a set of principles that provides a framework within which the laboratory studies are planned, performed, monitored, and archived and reported. We also have a question, why was GLP created? So this is because in the early 70s, FDA became aware of cases of poor laboratory practice all over the United States. They discovered a lot fraudulent activities and a lot of poor lab practices. Examples of some of these poor lab practices found were the equipment not being calibrated to standard form and therefore giving wrong measurements. And the next is incorrect or inaccurate accounts of the actual lab study. And the third is due to inadequate test systems. Purpose of the GLPs. The GLP is to certify that every step of the analysis is valid or not, and they should assure the quality and integrity of data submitted to FDA in support of the safety of regulated products. The GLP have heavy emphasis on data record and specimen retention. The good laboratory practices some of the principles. So there are 10 principles which the laboratory follows. The first one is test facility, organization and personnel. And second is quality assurance program, that is QAP. Then the facilities. Fourth one is apparatus, materials and the reagents. Fifth is test systems. Sixth is test and reference substances. The seventh one is standard operating procedure, that is SOP. And eighth is performance of the study. And ninth is reporting of study results. The last one is storage and retention of records and materials. Now we will see all the 10 principles one by one. So we will see the first one now, that is test facility, organization and personnel. Management responsibilities. Test facility management should ensure that the principles of good laboratory practices are compiled within the test facility. At minimum, it should have ensure that qualified personnel, appropriate facilities, equipment and materials are available, and should maintain a record of the qualifications, training, experience and a job description for each professional and technical individual. And it should ensure that personnel clearly understands the functions they are to perform and where necessary provide training for these functions. And it should ensure that health and safety precautions are applied according to national or international regulations. Then it should ensure that appropriate standard operating procedures are established and it is followed. And it also should ensure that there is a quality assurance program with designated personnel. Ensure that amendments to the study plan are agreed upon documented. And maintain copies of all study plans. Next, we have to maintain a historical file of all standard operating procedures. Then, for each study, ensure that a sufficient number of personnel is available for its timely and proper conduct. For each study, designate an individual with the appropriate qualification, training and experience, and we should also ensure that an individual is identified with the responsible for the management of the archives. Next is study director's responsibility. The study director has the responsibility of the overall conduct of the study and for its report. These responsibilities should include, but not a limit to, the following functions: should agree to the study plan, ensure that the procedures specified in the study plan are followed, and that authorization for any modification is obtained and it is documented together with the reasons for them, and it should ensure that all data generated are fully documented and it is recorded, and the sign and date the final report is indicate acceptance of responsibility for the validity of the data and to confirm compliance with the principles of good laboratory practice. And it should ensure that after termination of the study plan, the final report, the raw data and supporting materials are transferred to the archives. The next one is personal responsibility. The person should exercise a safe working practice. The chemicals should be handled with a suitable caution. The person should exercise health precautions to minimize risk of themselves and to ensure the integrity of the study. The person known to have a health or medical condition that is likely to have an adverse effect on the study should be excluded from operations that may affect the study. The second good laboratory practice is quality assurance program. So the quality assurance program, when you want to see it in general, the test facilities should have a documented quality assurance program to ensure that studies performed are in a compliance with the principles of good laboratory practice. The quality assurance program should be carried out by an individual or by individual designated by and directly responsible to management and who are familiar with the test procedures. The next is the individual should not be involved in the conduct of the study being assured, and the individual should report and findings in writing directly to the management and to the study director. So coming on for the responsibility of the quality assurance personnel, the responsibilities include ascertain that the study plan and standard operating procedures are available to personnel conducting the study, and we should ensure that the study plan and standard operating procedures are followed by the periodic inspections of the test facility by auditing the study in process, and the records for such procedures should be retained. Promptly report to the management and the study director unauthorized diversions from the study plan and from standard operating procedures. And we have to review the final reports to confirm that the materials, the procedures and the observations are accurately described, and that the reports which is given in the form of the results accurately reflect the raw data of the study. Next, we have to prepare and sign an assignment to be included with the final report. The third good laboratory practice is facilities. So we'll see first test system facilities. The test system facility should have a sufficient number of rooms or areas to ensure the isolation of the test system and the isolation of the individual products. And next is the suitable facilities should be available for the diagnosis, treatment and control of the disease. And there should be a storage areas as needed for supplies and for the equipments. Storage areas should be separated from areas which is from housing the test system and should be adequately protected against the infection and contamination. And refrigeration should also be provided. And coming on for the next, it is facilities for handling test and reference substance. To prevent contamination or mix-ups, there should be a separate area for receipt and storage of the test and reference substances, and mixing of the test substances with the vehicle. And the storage areas for the test substances should be separate from areas housing the rest system and should be adequate to prevent intensity, concentration, purity, stability, and ensure safe storage of hazardous substances. And the third one is archive facilities. Space should be provided for archives for the storage and retrieval of the raw data, reports, samples, and even for the specimens. And coming on for the last one, it is the waste disposal. The handling and disposal of waste should be carried out in such a way as not to jeopardize and integrity of the studies in progress. The handling and disposal of the waste generated during the performance of the study should be carried out in a manner which is consistent with regulatory requirements. This should include provision of an appropriate collection, storage, and the disposal facilities, decontamination and transportation procedures, and the maintenance of records related to the activities. The fourth good laboratory practice is apparatus, materials, and reagents. So we'll see first apparatus. The apparatus used for the generation of the data and for controlling environmental factors which is relevant to the study should be suitably located, and it should be in a very well designed and adequate capacity. The apparatus used in the study should be periodically inspected, cleaned, maintained, and calibrated according to the standard operating procedures. The records of procedure should be maintained. So coming on for the material, the apparatus and materials used in the study should not interfere with the test systems. And next are the reagents. The reagents should be labeled and indicate the source, identity, the concentration, and stability information, and should include the preparation date, the expiry date, and the specific storage instructions. The fifth good laboratory practice is test system. So we'll see the physical and chemical. Apparatus used for the generation of physical and chemical data should be suitably located and of appropriate design and adequate capacity. The reference substance should be used to assist in ensuring the integrity of the physical and chemical test system. So next, coming on for the biological, the proper condition should be established and maintained for the housing, handling, and care for animals, the plants, the microbial, as well as other cellular and subcellular systems in order to ensure the quality of the data. In addition, conditions should comply with the appropriate national regulatory requirements for the import, collection, care, and use of animals, plants, microbial, as well as other cellular and subcellular systems. The newly received animals and the plant test systems should be isolated until their health status have been evaluated. And records for source, date of arrival, and arrival condition should be maintained. The animal, plant, microbial, and cellular test system should be kept in an test environment for an adequate period before the study is initiated. All information needed to properly identify the test system should appear on the housing or even on the containers. the diagnosis and treatment of any disease before or during a study should be recorded. The next good laboratory practice is test and reference substance. So we'll see the first: receipt, handling, sampling and storage. Records including the substance characterization, date of receipt, quantities received and used in the study should be maintained. The handling, sampling and storage procedure should be identified in order that the homogeneity and the stability is assured without any contamination. The storage containers should carry identification information, the date of expiry and specific storage instructions. So coming on for the characterization, each test and reference substance should be identified. For each study, the identity including the batch number, the purity, composition, concentration or other characterization should define each batch of the test or reference substance should be known. The stability of the test and reference substance under conditions of storage should be known for all studies. And the stability of the test and the reference condition under the test condition should be known for all the studies. If the test substance is administered in a vehicle, the standard operating procedure should be established for testing the stability of the test substance in that vehicle. A sample for analytical purposes for each batch of the test should be retained for the studies in which the test substance is tested longer than 4 weeks. The next one is standard operating procedure. A test facility should have written standard operating procedures approved by management that are intended to ensure the quality and integrity of the data generated in the course of the study. Each separate laboratory unit should have immediately available standard operating procedures relevant to the activities being performed within. Published textbooks, articles and manuals may be used as supplements to these standard operating procedures. So when you take the application, the standard operating procedures should be available for but not limited to the following categories of the laboratory activities. So we'll see one, that is test and reference substance. The receipt, the identification, labeling, handling, sampling and also the storage. So when I come for the apparatus and reagent, the apparatus and reagents has to be in use, the maintenance, cleaning, calibration of measuring apparatus and environmental control equipments and the preparation of the reagents. Then third is record keeping, reporting, storage and retrieval. Here the coding of the studies, data collection, preparation of reports, indexing system, handling of data including the usage of computerized data systems. Next is test system. And the next is quality assurance procedures. So operation of quality assurance personnel in performing and reporting study, audits, inspections and final study report reviews. Then coming on for the last application, it is health and safety precautions. The eighth one is performance of the study, that is study plan. For each study, a plan should exist in a written form prior to initiation of the study. The study plan should be retained as raw data. All changes, the modifications or revisions of the study plan as agreed to be by the study director, including justifications, should be documented, signed and dated by the study director and maintained with the study plan. Content of the study plan should have identification of the study, the test and the reference substance, and the information concerning the sponsor and the test facility which is available, the dates, the test methods, issues, and the records. So coming on for the conduct of the study, a unique identification should be given to each study. All items concerning this study should carry their identification. The study should be conducted in accordance with the study plan. All data generated during the conduct of the study should be recorded directly, promptly, accurately, and by the individual entering the data. These entries should be signed or initialed and dated. Any change in the raw data should be made as and not to be abuse the previous entry and should indicate the reason, if necessary, for change, and should be identified by date and signed by the individual making the change. The data generated at the direct computer input should be identified at the time of the data input by the individuals responsible for direct data entries, and the correction should be entered separately by the reason for change with the date and the identity of the individual making the change. The ninth good laboratory practices, content of the final report. The final report should include but not be limited to the following information. The first one is identification of the study, the test and reference substance. Then information concerning the test facility, that is name and address, name of the study director, name of the other principal personnel having contributed reports to the final report. Next is dates, the statement, description of the materials and test methods, then results, and finally storage. The last good laboratory practice is storage and retention of the records and materials. So coming on for the storage should contain the study plans, the raw data, the final reports, the reports of laboratory inspections and study audits performed according to the quality assurance programs, then the sample and the specimens. Then coming on for the retention, the following should be retained for the period specified by the appropriate authorities. That is the study plan, raw data, samples, specimens, and the final report of each study. The records of all inspections and audits performed by the quality assurance program. Then the summary of the qualifications, training, experience, and job descriptions of the personnel. The records and reports of the maintenance and calibration of the equipments. The historical file of standard operating procedures. The samples and specimen should be retained only as long as the quality of the preparation permits evaluation. In this topic, we have seen what is good laboratory practice, the definition of GLP, and why GLP was created, and what is the purpose of the GLP, and what are all the good laboratory practices and the principles. Thank you. Thank you. Thank you.